Today we're going to be talking about bacterial transduction. And bacterial transduction is a process by which one bacteria gives another bacteria a piece of genetic information. And this piece of genetic information is going to allow the recipient bacteria to do something that it otherwise couldn't have done. It might impart antibiotic resistance. It might give the bacteria the ability to use a nutritional source that it couldn't use before. It might give that bacteria the ability to make a toxin that it otherwise wouldn't have been able to make. In any case, what's going to happen is this recipient bacteria is going to gain the ability to do something it couldn't do before. And that is going to give it a competitive advantage over its fellow bacteria and also other species of bacteria. So let's go ahead and talk about bacterial transduction. So bacterial transduction is going to start with a bacteria. And let's go ahead and draw the chromosome of this bacteria. So there it is right there. And bacterial transduction is going to involve a bacteriophage, a virus that infects a bacteria. So I'm going to go ahead and draw that. So there's my virus, my bacteriophage, and it's infecting the bacteria. And let's go ahead and draw a piece of DNA. There it is. The DNA is in red. It's inside the bacteriophage there. And once this virus attaches to the bacteria, that virus is going to inject that piece of DNA into the recipient cell. And this is where our next diagram comes in. Let's go ahead and draw this. We're going to draw the empty bacteriophage, still attached. There it is. And here's the viral DNA, and the viral DNA begins to replicate. And the host cell DNA actually busts apart into little pieces. Not only does the viral DNA in red here begin to replicate, but it also contains information to make viral proteins. And these viral proteins are going to get made, and inside the capsid, that's the head of this virus, the capsid, we're going to put viral DNA. But every now and then a mistake is made. And instead of stuffing a piece of viral DNA into this capsid, a piece of bacterial DNA accidentally gets stuffed into this capsid. And that might happen just a fraction of 1%. But because we're going to produce hundreds to thousands of viruses in this cell, that rare mistake is going to occur a couple times. Now, let's go ahead and go to the next diagram. So there's our bacteria. And I'm going to show fully formed bacteriophages in here. So there they are. I'm just going to draw a couple. There would be hundreds to thousands, but we'll just draw maybe 4 of them. And we'll draw one over here. There they are. And as you'll notice, they all have the viral DNA. However, once in a while, one contains a piece of bacterial DNA by pure chance, by accident. And when there's enough viruses created inside this bacterial cell, it's literally going to burst open. So I'm going to show that bacterial cell bursting open. There it is. And these viruses are going to come out of here, and they're going to infect nearby cells, and the process is going to continue again. But something different is going to happen with this virus right here, because it's not carrying viral DNA, it's carrying bacterial DNA. So I'm going to draw that virus coming out of here, and let's go up here so we can make our diagram high enough everybody can see. And I'm going to go ahead and draw another bacteria. And I'm going to show this other bacteria with a piece of green DNA, just to show that it's a different bacteria than this one here. And this virus, this bacteriophage, is going to go ahead and it's going to attach to this cell, and it's going to inject its DNA into that bacterial cell. Now, unlike viral DNA that's going to cause this bacterial chromosome to break apart and make more viral proteins and viral DNA, this bacterial DNA is a new piece of DNA. It might contain, again, a gene for antibiotic resistance, or a gene to break down a nutrient, or a gene for a toxin. And this bacteria, in a sense, has to decide, does it want that new piece of information, or doesn't it? If the bacteria decides that it wants that new piece of information, then a cut is going to be made between the nucleotides. We're going to break apart the nucleotides that connect that DNA together, and this piece of DNA is going to be inserted, there it is, into the host cell chromosome, into the recipient's DNA. And when that happens, this bacterial cell is now going to have the ability to do something that it couldn't have done before. And that's going to be it for bacterial transduction.